Okay, welcome to our chapter on cell division. Now, there are a couple things I just want to review with you before we move on because you need to have a basic understanding of chromosomes and parts of the cell before we continue with this chapter. So again, a cell is the basic unit of all living matter, and adult humans have over 10 trillion cells. The cytoplasm is the substance of a cell outside of the nucleus where all of the organelles float around. The nucleus of a cell is the brain. It contains the chromosomes, or the genetic material used to give the cell instructions. And the chromosomes, again, are made of DNA. There are 46 in each human body cell nucleus. 23 pairs because you get 23 from your mother and 23 from your father, and those match up in pairs to create 46. And again, those are made of DNA, which codes for all of the processes that the cell does. So we're going to talk about some chromosome terminology because this whole unit is going to be talking about chromosomes and how chromosomes divide during the cell cycle. So we need to get some basics of chromosomes here. Chromosomes are made of DNA wound around histones like spools of thread. So that sounds complicated, but it's really not. Chromosomes are these X-shaped structures over here. This is a big X-shaped structure, is a whole chromosome. And it's made of DNA. We'll get more into DNA in another unit, but right now all you need to know is that DNA is coding for all of the functions of the cell. They're the blueprints or genetic instructions. So here, I'm sure that you've seen a picture of DNA before, double helix, and I guess we did talk about it a little bit at the beginning of the year, but DNA is a double helix, and it's wound, if we get, this is a close-up here, if we get wind it up around here and make it even smaller and smaller here, around histones here, it winds around little histone proteins like spools of thread until it condenses into this X-shaped chromosome. A chromosome is made of two sister chromatids attached at a centromere. So here's one chromatid, one half of the chromosome, and this is the other chromatid. Sometimes we call them sister chromatids because they have matching genes on them, and they are attached by a centromere at the middle. Because the chromosomes are essential for the cell to function, specific processes are in place to sort them when a cell grows and divides. So that's why I'm saying that this whole unit is going to be about chromosomes and how they divide, because without those special processes for dividing the chromosomes, a cell would not be able to complete all the functions that it needs to. So there are very specific processes in place. And when that cell divides, that's called the cell cycle. So there are two general periods during the cell cycle, and this is, you'd look at this cell cycle just like you'd look at a clock, and it continues on over and over and over again. And the first period is cell growth, and that's going to be what's happening in this yellow area here. Orange. The orange area is all cell growth. Cell division occurs in this tiny little chunk of time in the M area that's called mitosis. So as I said, most of the cell cycle is spent in the growth phase, not mitosis. Think of the cell moving through the cell cycle like a clock hand moving around a clock. Stages don't happen independently from each other. It's a continual process as cells move from one phase to the next. So we are going to break down some of these phases here. We have G1, you have S, you have G2, and then there's a bunch of phases here in mitosis. All of those are going to be an ongoing, continual process. They're not choppy, stopping and starting. So let's start with interphase. Interphase is the growth period of the cell. So this is when the cell is getting larger, getting ready to divide, but not dividing yet. In interphase, the cell grows and carries on metabolism. So all the organelles are doing all of the processes and functions that we talked about in the cell organelle unit. chromosomes are duplicated. And so let's talk about why that's important. Why do the chromosomes need to be duplicated at all? If the cell divided without duplicating the chromosomes, then when you had two cells at the end of the cell division process, each new cell would only have half of the necessary genetic material. And that would be detrimental for the cell because there's no way that a cell could survive with half of the genetic material because it just wouldn't have all the instructions that it needs to carry on metabolism. So rather than having the chromosomes divide in half and have half left every time the cell divides, we need to duplicate the chromosomes so that when there are two new cells from the one cell, both of the new cells have the complete number of chromosomes. Okay, so there are three parts to interphase. An interphase is this whole orange section here, whole orange section. And as you can see, it's divided into three parts. G1, where the cell grows and duplicates its contents other than chromosomes, so it's going to create duplicates of all of the organelles so that the organelles don't end up getting divided in half. So there'll be an appropriate number of mitochondria, an appropriate number of lysosomes in the new cells. And sometimes, I wasn't going to mention it here, but sometimes the cell will actually jump out of this G1 phase and go through a G0 phase, and that's really just like it stops doing anything towards division. It's just growing and doing its normal business. In the S phase of interphase here, the cell is copying its chromosomes. We just talked about why that's important. And in G2, the cell grows some more and checks the duplicated chromosomes because you don't want any errors in there. We talk about this a little bit later, but errors in chromosomes or DNA are called mutations. So if a cell ends up creating a new cell with a mutation in it, that could create havoc for the organism. Okay, so after interphase, we enter this tiny little period called mitosis where the cell divides. And even though it's a small portion of time in the cell cycle, mitosis has some very specific parts to it, so we're going to spend extra time here. So mitosis is the period of division in somatic cells. Somatic cells are body cells. And the division of chromosomes occurs first, and then the division of cytoplasm and all the other organelles. One parent cell becomes two identical daughter cells. So we start with one cell, and at the end of the process, we've divided into two different cells. But those cells are actually identical to each other because the whole process of mitosis relies on the fact that we are making the same cells over and over again. So here are the different phases of mitosis. There are four main phases which are responsible for splitting the chromosomes: prophase, metaphase, anaphase, and telophase. And then additionally, after that, after the chromosomes are split, in cytokinesis, the cytoplasm will be split. Okay, so let's take a look first at prophase. And you'll be able to see on this picture here, this is our cell cycle going around here. I know it's a slightly different picture of the cell cycle here, but this arrow shows where we are. We are right here in prophase at the beginning of mitosis, and this is what the cell is going to look like in prophase. Strings of chromatin coil into chromosomes. Okay, so chromatin is like a long, stringy DNA strand, and it coils up, as we talked about at the beginning here, into chromosomes around the histones, and it condenses into those X-shaped structures. So the DNA does not always look like this. Sometimes it looks like long strings, but when it needs to divide, rather than dividing out long strings, it condenses so it doesn't get tangled. Okay, the chromosomes, as we mentioned, have two halves called sister chromatids, and both those halves are exactly the same. and they are connected in the middle by a centromere. Also in prophase here, we have a nuclear envelope that is disappearing. If you remember from the cellular organelle chapter, there is a nuclear envelope protecting the DNA in the nucleus, and it allows some things to move in and out, but mostly it protects the DNA. And this nuclear envelope needs to disappear as the cell divides because the chromosomes are going to have to move to create two new cells. So the nuclear envelope disappears. Centrioles, which are these orange structures at the end, those migrate to the poles or the ends of the cell. Usually they're kind of just floating around in the middle, but they actually move to the ends of the cell. And spindle fibers, which you can think of as long threads, form in between these centrioles. Okay, those are the blue ones, blue fibers there. All right, as we move on to metaphase, the chromosomes attach by their centromeres to the spindle fibers. So you can see here, the centromere in the middle of the chromosome attaches one side and the other side to spindle fibers. Chromosomes line up along the middle of the cell. So that helps you remember what's happening in metaphase. I always tell students remember middle, meta, and that should help you keep some of your phases straight. Each chromatid is attached to its own spindle fiber. All right, in anaphase, as we move around the cell cycle here, now we're in anaphase. The cell is elongating, and the centromeres, the center of the chromosomes, is splitting in half. Each chromatid is pulled to the opposite end of the cell. So they are moving away from each other. Good way to remember anaphase starts with A, away. They're pulling apart or away from each other. And then in telophase, those chromatids reach the ends or reach the poles. And the chromosomes begin to unwind again, and all the processes from prophase start to reverse. So they unwind back into chromatin, or that long tangly DNA, and the nuclear envelope begins to reappear. You can see that dotted line there. Okay. The centrioles and the spindle fibers will eventually go back to their original position as well. Okay, so now we've divided the chromosomes, and we're on to cytokinesis, or the division of the cytoplasm. And this is a little bit different in plant and animal cells. All the other processes in mitosis occur the same way in plant cells and animal cells. But in cytokinesis, animal cells have a plasma membrane that pinches along the center and forms two daughter cells. So that's what you can see happening right here. It's pinching in half. And in plant cells, because they have a cell wall, they have a slightly different process. A cell plate forms in the center, right in the middle here, and then it grows and grows and grows until it's an extension of the cell wall. And that's how plant cells go through cytokinesis. Okay, so just as a summary or an overview here, in mitosis, one parent cell creates two daughter cells that are identical to each other and identical to the parent cell. So the whole goal is to create two cells that are exactly like the first cell. Reasons that mitosis occurs. So when is this happening in your body? First of all, anytime an organism is growing, the cells don't just get bigger, they divide and there are more and more and more cells. So anytime an organism is growing, it's going through mitosis. If there's healing taking place, that means if you got a cut and after the scab forms, you need to replace and reform some of those skin cells. So that healing process, the creation of a scar, all of those require mitosis. And anytime old cells are being replaced, you need mitosis to replace those cells and create new cells. So like when you're taking a shower, or you take off a sweater and you notice that there's some dead skin on the on the sleeves or whatever because your skin's dry, those are cells sloughing off, and your body is constantly replacing those over and over and over on a daily basis. Okay, so now we're going to move on to our other process for cell division, the mitosis process occurs in all of your body cells. The next process here is for sexual reproduction only. So this is going to occur only in your sex cells. And this process is called meiosis. Okay, so meiosis, which I pronounce meiosis, but some people pronounce it meiosis, either one is really correct, is the cell division that occurs to produce sex cells. We call sex cells gametes, and those are either going to be eggs or ovum or ova in females or sperm in males. Those are the only cells that are going through this meiosis process. Why must meiosis occur in these cells rather than mitosis? So we're talking about sexual reproduction here. The sex cells have a different process than all the rest of the trillions of body cells. Because sex cells were designed to be combined with another sex cell, eggs combined with sperm. Therefore, if these cells were created by mitosis, when they combined with another sex cell, the zygote, which is that tiny little baby cell that would form, would have double the correct number of chromosomes. You wouldn't think that that's necessarily a bad problem to have, but just like losing half of the instructions if the chromosomes didn't duplicate in mitosis, getting double the number of chromosomes that you're supposed to have is also detrimental to a cell. So we can't have that. So the whole process of meiosis is to actually reduce the number of chromosomes by half. So meiosis begins the same way as mitosis, with the chromosomes duplicating. Unlike mitosis, though, there are two periods of cell division because, as I just said, the chromosome number needs to be reduced by half. So if we start with human cells, there are 46 chromosomes. If we start with 46 chromosomes in the parent cell, we can't have 46 in the egg cell and 46 in the sperm cell. We have to get all the way down to 23 in the egg cell and 23 in the sperm cell so that when they combine, there's a total of 46. So there are two periods of cell division here. We have Meiosis I and then Meiosis II. And then by the end of Meiosis II, you have 4 cells, and each of those 4 cells has half the original number of chromosomes. Okay, so in Meiosis I, homologous chromosomes separate. And your homologous chromosomes are the ones that match, one from the mother and one from the father. And these, in this picture here, don't match exactly, and we'll talk about why that is. In Meiosis II, duplicated chromosomes separate, and that's going to look just like mitosis, where you have a chromosome that's going to divide in half, and then you have sister chromatids at the end. So we'll go through all the stages here. But the end result, because of this switching of colors here, which again we're going to talk about real soon, the end result of meiosis is 4 different cells with half the number of chromosomes. And having different cells at the end of meiosis is very good. Okay, so parent cells in meiosis begin with what we call tetrads. A tetrad is a pair of matching homologous chromosomes. So homologous, homo, we talked about earlier in the year. We were talking about homogeneous mixtures, which were the same throughout. Homo means same. So these are matching or same chromosomes. So for example, this would be like the 16th chromosome from your mom and the 16th chromosome from your dad. And they are organized by length, and you will look in depth at that in some future units. But a tetrad, for now, is a set of homologous chromosomes. Okay, so here is one of the special things that happens in meiosis that does not happen in mitosis. As the parent cell is preparing to divide for the first time in Prophase I of Meiosis I, the homologous chromosomes in a tetrad can cross over. So here you have your homologous chromosomes, and then two of the arms here, one arm or two arms, it depends on how they tangle exactly, allows the arms here of the chromosome to be broken off and switched. And this process allows genes to be traded between homologous chromosomes. So these genes actually got traded one chromosome to the other. And it's not an exact science. Sometimes one gene will get traded, sometimes another one, and it just depends on how they all overlap. But that process is called crossing over. And crossing over is why siblings have the same parents' chromosomes, because they have the same parents. So I have a sister, my sister and I both got the same set of chromosomes from our parents, but a slightly different combination of genes, because depending on how those chromosomes crossed over, we ended up with a different set, mixed and matched of genes. And that makes each cell that is created, each egg cell or each sperm cell, slightly different. So that each baby, when it produces a baby, is going to be slightly different. Okay, so in Meiosis I, let's take a look at the phases of Meiosis I. Starting with Prophase I. Homologous chromosomes pair up into tetrads, as we just mentioned, and they cross over. In Metaphase I, homologous chromosomes line up in pairs along the middle of the cell. In Anaphase I, homologous chromosomes separate. So now you have the 16th chromosome and the 16th chromosome, each going their own separate way. In Telophase I, homologous chromosomes reach the end. So now you have two cells, each with one set of chromosomes. And then Meiosis II is going to run just like mitosis. In Prophase II, the nuclear envelope disappears. In Metaphase II, the chromosomes line up along the middle of the cell. In Anaphase II, the sister chromatids separate. In Telophase II, the sister chromatids reach the ends of the cell and the nuclear envelope reappears. And then, as you can see, in each of these cells, you have slightly different chromatids left because of the crossing over. So now we have 4 cells that have half the number of chromosomes, and they are all slightly different. So if we compare mitosis and meiosis here, let's take a look at mitosis. We start with one parent cell. It creates 2 identical daughter cells. They're identical to each other and to the parent cell. Daughter cells have the same number of chromosomes as the parent cell. It is used for healing, growth, and replacing dead cells, and it's a type of asexual reproduction. In other words, one organism is producing more over and over again all by itself. In meiosis, we start with one parent cell, and at the end we have 4 different daughter cells. Not only are they different from the parent cell, but they're also different from each other. The daughter cells have half the number of chromosomes as the parent cell, and it's only used for creating sex cells or gametes, eggs and sperm. And it prepares the organism for sexual reproduction with another organism. By reducing that chromosome number by half, that organism is now prepared to match up gametes with another organism in sexual reproduction. Okay, so that's all well and good, but how does the cell cycle affect my life? Well, besides the obvious formation of cells, healing, growth, and sexual reproduction, all things that are important for your life, there are other ways that the cell cycle might be important to you, or things that you need to know about that you may encounter throughout your life. First, when the genes that control the cell cycle are mutated, it's like a clock. There's a process, there's a set of, in a clock there's a set of gears, in a cell there's a set of genes that control how fast that moves. If those are mutated, cells can begin to divide at an uncontrolled rate. And this is how tumors form. Tumors are a growth of cells, and those cells have grown uncontrolled. And that's how cancer can progress through someone's body. So, for sure, cell cycle is pertinent to your life if you know anyone that has cancer. Secondly, if chromosomes are not evenly divided between the new cells, genetic abnormalities can result. So if this occurs in the gametes during the process of meiosis and the chromosomes are not evenly divided, it can cause birth defects or miscarriages. So when a baby does not have the right number of chromosomes, or close to the right number of chromosomes, if we're talking about way off the correct number of chromosomes, then it's going to be really difficult for that baby to survive, and that's one of the reasons that miscarriages can happen. So, see, understanding the cell cycle can lead to great advancements in modern medicine. The more you know about the cell cycle, the more you can relate to medicine and current events. And that's where we'll stop for this week.